Good afternoon, everyone. In this talk, I would like to give you an overview and an outlook on emerging fMRI methods for improved neuronal specificity for human imaging. As an overview of this talk, first, I'll go over the classic T2-STAR-BO sequence, which is the workhorse sequence for functional MRI, describe to you its main limitation in terms of limited neuronal specificity, then talk to you about some alternative contrast encoding mechanism that can help us overcome this issue, particularly with T2-BO, CBP, and CBF, while concentrate on looking at work that has been done at ultra-high field on human imaging, where there is sufficient SNR to push to sub-millimeter resolution. Then I'll move on to talk about some of the exciting and emerging approach and trends in overcoming some of the drawbacks that has plagued some of these alternative contrast encoding mechanisms. And I will conclude with some outlook on how we can use these contrast mechanisms to move towards neuronal specific high spatial temporal resolution imaging with fMRI in the human brain. So first, T2-STAR-BO fMRI. This is the most widely used sequence, has high SNR and simple encoding. Over the last decade, there's been significant improvement through volumic encoding using SMS and 3D EPTI to allow this approach to be acquired at high spatial and temporal resolution. Example here at 1 millimeter isotropic whole brain acquired with a TR of just 3 seconds. With T2-STAR-BO, we can now robustly acquire data at sub-millimeter resolution. Here's an example from Polimini et al., where they're trying to activate an M logo onto the visual cortex. At high spatial resolution, you can start examining things at different cortical depth with clear delineation of this M logo across these depths. However, there's also some artifact and contamination, particularly near the PL surface. And this has been the main limitation of T2-STAR-BO in terms of contamination from PL and ascending veins. To overcome this issue, people have worked quite a lot on T2-BO, which has been shown to be less sensitive to PL and ascending vein, particularly at ultra-high field. Here's some classic work by Isa Yakub showing that with spin echo EPI, he can get better visualization of ocular dominant column mapping compared to gradient echo EPI. However, for the spin echo EPI that Isa did, it's important that he uses a very short readout window to be able to get improved spatial specificity. This issue has been nicely highlighted by Gonce and Logothetis in their monkey study at 4.7 Tesla. Here, if you look at gradient echo EPI, you can see large PL vessel contamination. Moving to spin echo EPI with 4 shots, this contamination is still present because the EPI encoding window is still around 30 milliseconds. Therefore, you have a lot of T2-STAR waiting, particularly at high case-based frequency. Moving to 8-shot, things helps a bit further. And then getting to 16-shot, you can get rid of most of this bias and get the nice signal concentrated in the parenchyma as expected from neuronal signal. So with spin echo EPI, it's important to do segmentation and multi-shot, which is challenging, leading to susceptibility to motion and physiological noise, as well as long acquisition time and small field view coverage. Nonetheless, some really nice work has been done with T2-BO, particularly with 3D grace sequence, where with 3D grace, you do a slab excitation and refocusing in orthogonal planes, so you can do a zoom imaging with a reduced volume. This helps reduce the encoding burden, allowing you to have shorter EPI readout after each refocusing pulse so that you have less T2-STAR contamination. You've been able to get nice result with the zoom gray sequence in the visual cortex, for example, here at 0.7 millimeter ISO. Here's another example showing nice result that the gray sequence is not biased at the PL surface when compared to gradient echo, and you get this nice flat profile of activation across the V1 region. Here's more recent work in the motor area showing the nice double peak across the cortical depth as expected. The main drawback of grace has been in the limited slice coverage because of the limited number of refocusing pulses that can be applied, and the KC encoding that can be performed before the signal decays away. Variable flip angle has been used to try to overcome this issue, allowing for longer echo train with reduced C blurring. More recently, work has also been done on spatial temporal random encoding and constrained construction to allow for higher acceleration and improve SMR. Through these development, the field view of grays can be increased by fourfold along the slice direction, as well as achieving better SNR in the fMRI activation. In talking about spatial temporal encoding and reconstruction, this theme of method has also been applied to multi-shot EPI, particularly with the equiplanar time resolve imaging, or EPTI, that we've recently developed. This is a novel distortion-free multi-shot EPI approach where the encoding is performed in KT space. Each shot of EPTI try to cover a large section of KY using a highly undersampled spatial temporal Chi-P sampling pattern. Using a few of these shots, the whole KT space can be covered, and using a synergetic reconstruction, you can fill out this whole space nicely. With full KT space at each time point, then you have full K-space data so that you can extract out a distortion and blurring free image at that particular TE contrast point. So with the EPTI acquisition, you can then time resolve hundreds of different images across different TE throughout the whole readout. So this approach has been applied to fMRI to try to generate PLT2 ball with minimal T2 prime contamination. Here's an example in the visual cortex. With the EPTI approach, you can get multiple images across the readout. We're going to pick 3 of them. Here, one at the spin echo and a couple at gradient echo weighted points. Then look at the time series of this data and plot the activation across cortical depth for these time series. As you can see, for the gradient echo weighted acquisition, you have influence of large vein, whereas in the spin echo, the activation across cortical depth remained pretty flat. There's also a clear classic trade-off between sensitivity versus specificity as you move from gradient echo to spin echo. We can replot this in a normalized plot where we normalize with the gradient echo 1 around the center depth so that we can kind of see things a bit better in terms of the slope differences for different cases. Moreover, with the EPTI approach, you also get the full KT data, and this allows us the opportunity to extract out data of what it would have looked like if we were to acquire the standard multi-shot EPI approach. So we extract 2 sets of data out of this full KT data. One is a multi-shot spin echo EPI with an echo train length of 36 milliseconds, and a second one with an echo train length of 24 milliseconds. From this images and their time series, we can then look at the activation across cortical depth, where we can see that with shorter and shorter echo train length, you get decreasing T2 star contribution, getting closer and closer to this flat profile that we expect. So now, having talked quite a bit about T2 bow, let's move to CBB, where the CBB work got started in animal models with Mayan contrast agent that was shown to be to provide capillary specificity. In terms of human imaging without contrast agent, a number of approaches has been developed, particularly with the vascular space occupancy or the VASO approach, which has really taken off in recent years. So with VASO, you perform an RF inversion and wait for a certain TI to image, where the TI is chosen to be a point at which the blood signal is null. During functional activation, the blood volume will increase, causing an increase in the vascular space of this null signal within each given voxel. This will reduce the overall signal in that voxel as CBV increase, allowing you to track CBV with a reduction in signal. A couple of innovative extensions has been made to VASO to make it work really well. First of which is to replace a non-selective inversion with a slab selective inversion. This allows fresh blood to enter in at each TR, making the signal of blood much more different than the gray matter signal. This is really helpful at ultra high field, where the gray matter signal and the blood signal has very similar T1. Having this differential at the start will help differentiate the signal at the TI point. An additional also an additional slice saturation pulse has also been added near the end of the TR of an acquisition. This also increased the gray matter signal by another tenfold, which has been really helpful. Furthermore, with this slice saturation, you can also acquire second EPI data that has ball weighting and use this as a control to remove ball contamination from your vasal acquisition nicely. So with this size-saturated slab selective vasal method, you can get nice result. You get both simultaneous bowl and vasal, where bowl has a lot of bias and vasal doesn't at the surface. Nonetheless, with this blood nulling method, you can only image close to that blood nulling point, limited the number of slice coverage that you can perform. A number of approach has been proposed to try to overcome this issue, most recently by moving away from the blood nulling contrast to more of a T1 weighted contrast to get CPV, particularly using the magic vasal approach, which acquire alternative T1 weighting with variable flip angle at different TRs. So in the odd TR, a large flip angle is used so that there's a difference between gray matter and blood signal, having CPV contrast. And in the even TR, data is acquired with a smaller flip angle so that there's no difference in the MC of gray matter and blood, and you get bowl contrast that you can use to remove bowl contamination from the vasal acquisition. So this sequence provide increased flexibility in encoding because you're no longer limited to image close to the blood nulling point. So you can have a very long TR, for example, where you acquire high spatial resolution with large brain coverage, or you can go for a short TR with a small coverage, but with high temporal resolution. Here's an example of close to whole brain 0.8 millimeter isotropic magic vasal acquisition with good activation sensitivity. Here's another case with limited coverage, but at a very high temporal resolution, showing that you can use this to demonstrate that CVV has a delayed return to baseline compared to bowl. So vasal development has been great. However, it still has about half the SNR of T2 star bowl. So approach via spiral has been proposed to try to boost its SNR through acquiring data at much shorter TE with less T2 star decay. In this example here, with spiral, you gain more than 2-4 SNR compared to an equivalent EPI acquisition. As things progress further to sub-millimeter voxel size, I feel that it will be important to be utilizing the expanded signal model for off-resonant and trajectory correction. Some really nice work here by the ETH group with that field probe to be able to get good sub-millimeter spiral images. Lastly, I'll talk a little bit about serial blood flow through the ASL method, which has potentially more, can provide potentially more specificity compared to vasal bowl CBV, which has some remaining sensitivity to arterial blood. However, ASL has about 10 to 20% SNR of T2 star bowl, so it's challenging. Nonetheless, some promising result has been acquired at ultra high field, 0.9 millimeter here ASL with 3D PI, as well as the utilization of spiral to increase its SNR. Moreover, spatial temporal encoding and reconstruction has also been applied to 3D Grace ASL. In this case, in each acquisition, you do a highly undersampled 2D K.P sampling and do a complementary sampling across the different TRs and utilizing a joint reconstruction across TR to get good reconstruction. So with this approach, you can get improved perfusion map with reduced sensitivity to motion and physiological fluctuation. The approach has not quite been extended to fMRI yet, but I think this would be an interesting direction to push forward in terms of incorporating example like low rank constraint that has already been done for task-based functional MRI by Mark Chu and his group at Oxford, for example. So in this talk, hopefully I've shown you some promising approach to increase spatial specificity of fMRI, moving away from T2 star to T2 and T1 based contrast. But this comes at a cost, particularly reducing sensitivity which can be mitigated through better volumic encoding, spiral acquisition, and clever sequence design. With these contrast encoding mechanism, you also have more interaction between contrast and image encoding that you have to manage and optimize. In particular, with T2 bowl contamination, requiring you to do segmentation or zoom, as well as trying spatial temporal encoding and acquiring an extra control bowl acquisition, as an example. Another issue that you have to deal with is in terms of constraint on achievable spatial temporal resolution and coverage that you can achieve with these methods due to the increase in encoding burden, extra time for contrast prep, as well as limited acquisition window. A number of approaches is being proposed to fix this, including extended equitrain to variable flip angle, spatial temporal encoding, and clever sequence design by moving away from blood nulling to T1 weighted sequence in magic basal, for example. So these emerging innovation and techniques have led to high spatial resolution, large coverage, and fast sampling for fMRI, but you can, with these approach, you can't achieve all these three things simultaneously yet. So we're probably still a factor of 3 to 5 away from, in terms of encoding efficiency, from getting to 0.5 millimeter whole brain acquisition with good temporal resolution. The direction to work on further still to get there would be combining some of these emerging ideas, which are complementary to provide additional gains, as well as looking into new innovation, particularly in the direction of spatial temporal encoding, which I think would be promising, as well as more sequence design and clever approaches in that direction. And lastly, relying on some of the exciting emerging hardware technology that has been coming out to really improve SNI encoding efficiency further as well. So with that, I'd like to thank you for your attention.